Hello everyone, welcome to my channel. The topic for today's discussion is Ramachandran Plot. It is one of the most important topics from your biochemistry syllabus. So let's get started. The phi and psi plot of the amino acid residues in a peptide is called Ramachandran plot. It was developed by Gopala Samudram Ramachandran, who was an Indian physicist, and he had given us this plot to study amino acid in proteins in the year 1963. It involves plotting the phi values on the x-axis and psi values on the y-axis to predict the possible conformation of the peptide. The angle spectrum in each axis is from minus 180 to plus 180 degrees. Here we are mainly going to talk about torsion angles, that means dihedral angles which are formed by two planes. Okay, how do they rotate? The core regions, the allowed regions, the disallowed regions in the plot. And in this plot, where are we going to find alpha helices, beta sheets? Okay, so this plot basically shows secondary structure of protein. So what is this plot showing us? Shows secondary structures of protein, right? See, as you all know, peptide bond is also known as amide bond. It's a covalent bond. Okay, it is formed from condensation. That is, water is formed here, and it is usually found in trans conformation. Right? It has partial double bond due to resonance, and trans conformation is always more favored than cis conformation. Why is cis conformation less favorable? It's because of the steric hindrance between the functional groups attached to alpha carbon. Fine. The steric hindrance between the functional groups attached to C alpha is more in cis configuration. And in trans configuration, what happens? The two C alpha are not close by. Okay. See, the two torsion angles of the polypeptide chain, also called Ramachandran angles, these angles are also called Ramachandran angles, describe the rotations of the polypeptide backbone around the bonds between N C alpha and C alpha C. The angle here, that is phi, this angle is between this one, N and alpha carbon, NC alpha. And C alpha C also has an angle here which is known as psi. Okay. These two angles, that is phi and psi, they can rotate. Clear? They can rotate. They have a single bond nature. Whereas omega, omega is fixed. Right? It is fixed, hence no rotation. Its value is also from 0 to 180 degree, but there is a double bond character here. There is a double bond character, hence it can't rotate. Okay, fine. Now see, the Ramachandran plot provides an easy way to view the distribution of torsion angles of a protein structure. It also provides an overview of allowed and disallowed regions of torsion angle value, serving as an important factor in the assessment of the quality of protein three-dimensional structures. Okay, here actually we get to see three regions in the Ramachandran plot. We'll come to it. See, we get to see three regions in Ramachandran plot. What are they? The core region, then we get to see the allowed region, and then we get to see the disallowed region. Okay, we'll get back here. Now see, we have four quadrants here. Okay, there are four quadrants in the Ramachandran plot. See, there are four quadrants here. Now we'll first try to see the plus and minus values in the four quadrants. So this is your quadrant 1, this is quadrant 2, this is quadrant 3, and this is quadrant 4. So you have the four quadrants here. Okay. As discussed earlier, the value of phi is plotted on the x-axis and value of psi is plotted on the y-axis. Okay. The values ranges from, that also you know, the values ranges from minus 180 degrees to plus 180 degrees. So here we'll have minus 180 degrees and here we will have plus 180 degrees. Same for this ordinate as well. Plus 180 degrees here and minus 180 degrees here. Clear? Then see, the value of psi and phi here will be what? Value of psi and phi. Here you see the value of psi is plus, whereas the value of phi is negative. Because see here it's all negative, right? This whole part is negative. Here, what do we see for psi and phi? Tell here, what do we see for psi and phi? We see positive values for both. Both psi and phi is positive in quadrant 2. In quadrant 3, come here. We see both psi and phi values to be negative, and in quadrant 4, we see psi and phi values to be negative and positive. Okay, this is something very important and you must know. Clear? Okay. Now see, the Ramachandran plot helps us with determination of secondary structures of protein. We have just discussed this. Fine. Now we will study each quadrant here. The four quadrants which we have just now discussed. Quadrant 1, quadrant 2, quadrant 3 and quadrant 4. Okay. Now see, quadrant 1 shows a region where some conformations are allowed. This is where rare left-handed alpha helices lie. Fine. In quadrant 1, here, look at this. Somewhere here you have, I'll change the color, it will be easy. Wait. Okay. Somewhere here like this, you get to find the right twisted beta sheet. Twisted beta sheet. Okay. Here you have the parallel beta sheet. Okay. And somewhere here you have the anti-parallel beta sheet. If I write here, anti-parallel beta sheet. Okay. Somewhere here you will have collagen or collagen triple helix. Okay. Next, coming to quadrant 2. Quadrant 2 shows the biggest region in the graph. This region has the most favorable conformation of atoms. It shows the sterically allowed conformations for beta strands as well. Other than that, see, you also have the left-handed alpha helix here. You can see here. See? Fine. Next, we'll go to the third quadrant. The third quadrant, see what I was writing there is here. You can also see it. The third quadrant shows the next biggest region in the graph. This is where right-handed alpha helices are found. What is it? Right-handed alpha helices. So you'll have to remember each quadrant along with the particular types of amino acids found. So here you have the right-handed alpha helices. Next is the fourth quadrant. What do you have in the fourth quadrant? Has almost no outlined region. Important point. It has almost no outlined region. This conformation is disfavored due to steric clashes or steric hindrances. This conformation means what? Psi around minus 80 to 0 degrees and phi around 0 to 180 degrees is a disfavored zone due to steric hindrances. All right. So this is how the Ramachandran plot looks, and this is how we decipher the plot. This is how we interpret the plot. Okay. In this, like in the disallowed region, for example, in the disallowed region, you get the you get glycine because they don't have steric hindrance. And why is it that they are not having steric hindrance? It is because they don't have side chain. Fine. This is an exceptional case actually. Glycine faces no hindrance and it is frequently seen in the plot. Whereas proline is not seen frequently, rather rarely. It has, if I see these things we write, glycine frequently seen in the plot. Right? Whereas proline not seen frequently in the plot. Why? Why is glycine seen so frequently in the plot? Because faces no hindrance. Since they have no side chain. Since no side chains. Remember this. The slide is. Other than this, proline is not seen frequently in the plot. Fine. What is the explanation? It has bulky R group. It has bulky R groups. Okay, side chain is connected to the protein backbone twice. Steric hindrance is more. Okay. Clear? Now, see, each quadrant has to be associated with the psi and phi values as well as the amino acids found. That is very important. So this is how we come to an end of our lecture. I hope this brief lecture was helpful to you. It was very simple and I have emphasized mainly on the torsion angle, the values of phi and psi at different quadrants, and you must remember, along with the phi and psi values in the different quadrants, you must remember where is the position of each of these types, like the antiparallel beta sheets, the parallel sheets, the right hand alpha helix, the left hand alpha helix. And remember, so far, whatever we have discussed was about L amino acids. If it is for D amino acid, it will be just the opposite because L amino acids and D amino acids are mirror images of each other. Okay, so these little points you need to be very careful. Another important point, just as a recapitulation, I'm telling you, peptide bonds or amide bonds, as we have studied, they form via dihydrolysis reactions, okay? And these are thermodynamically not favored. So it requires ATP. Clear? And these peptide bonds have a partial double bond character, which makes it rigid. As I told you, single bond character. favors rotation, double bond characters, or partial double bond characters doesn't favor that. So these little points before ending, I just tried to recall so that it becomes well seated in your head. So till then, thank you so much for joining me. Bye bye.